Hello and welcome everyone. Myself Mijanur Rahaman, and today my topic of discussion is mechanism of hormonal action. So in the previous video we have discussed about the hormonal action, and now we will discuss about the mechanism of the hormonal action. So hormone does not act on the target cell directly. We all of us know that. And it combines with the receptor to form hormone receptor complex, and this hormone receptor complex executes the hormonal action by any one of the following mechanism. There are three mechanisms known so far by which hormonal action mainly happens. First of all, by altering permeability of the cell membrane, by activating the intracellular enzyme, and by acting on genes. So there are three methods. One is by altering permeability of the cell membrane, number two by activating intracellular enzyme, and number three by acting on genes. First of all, by altering permeability of cell membrane. Now, neurotransmitter in the synapse or neuromuscular junction act by changing the permeability of postsynaptic membrane. So, in this method, what generally happens? Neurotransmitters in the synapse or neuromuscular junction act by changing the permeability of postsynaptic membrane. For example, in a neuromuscular junction, when an impulse or action potential reaches the action terminal of the motor nerve, acetylcholine is released from the vesicles, and acetylcholine increases the permeability of the postsynaptic membrane for sodium by opening the ligand gated sodium channel. So sodium ions enter the neuromuscular junction from ECF through the channels and cause the development of end plate potential. So what happens in a neuromuscular junction? Whenever any action potential or any impulse is generated and reaches to the action terminal of the motor nerve. So first of all, one impulse is coming, and with this is going to the motor nerve action terminal. Then what happens? Acetylcholine is released from the vesicles. Now when the acetylcholine is released, it is increases the permeability of the postsynaptic membrane for sodium, and when it increases the postsynaptic membrane permeability for sodium, it already increase and open the ligand gated sodium channel. When the ligand gated sodium channel is opened, then what happens? Sodium ions enter into the new NM junction from extracellular fluid through the channels and cause the development of end plate potential. Thus the end plate potential is generated. Now, second method. From this method, we have come to know the by altering permeability of cell membrane. So, in this method, the neurotransmitter acts. Then the second method, by activating the intracellular enzyme. The protein hormones and the catecholamines act by activating the intracellular enzymes. So, what are the components that act upon this type of mechanism? Number one, protein hormone, and number two, catecholamines acts by activating the intracellular enzymes. So, in this method, there are some messenger occurrence. First messenger, first of all, what is first messenger? The hormone which acts on the target cell is called the messenger or chemical mediator. It combines with the receptor and form hormone receptor complex. So whenever the hormone receptor complex is formed, then the first messenger is acting. So what the first messenger acting? The hormone receptor, the hormone which acts on target cell, it is called first messenger. So what hormone is known as the first messenger which is acting on the target cell or chemical mediator. It combines with the receptor and forms the hormone receptor complex or HR complex. Now second messenger. The hormone receptor complex activates the enzyme of the cell and causes the formation of another substances called second messenger. So hormone receptor complex activates the enzymes of the cell and causes the formation of another substances called second messenger or intracellular hormonal mediator. And the second messenger produces the effects of the hormone inside the cell. Protein hormones and the catecholamines act through the second messenger. Most common second messenger is cyclic AMP. So most common cyclic messenger is known as CAMP or cyclic AMP. So cyclic AMP, CAMP or cyclic adenosine 3' 5' monophosphate acts as a second messenger for protein hormones and catecholamines. So cyclic AMP or CAMP or cyclic adenosine 3' 5' is or 3' 5' monophosphate acts as a second messenger for the protein hormones and catecholamines. Then formation of CAMP and role of G proteins. G proteins or guanosine nucleotide binding proteins are the membrane proteins situated on the inner surface of the cell membrane. So what is G protein? G protein or guanosine nucleotide binding proteins are the membrane proteins which are situated on the inner surface of the cell membrane. Now these proteins play an important role in the formation of CAMP. Each G protein molecule is made up of trimeric or three subunits called alpha, beta and gamma. The G protein is made up of what? There are three subunits present in G subunit. One is alpha, one is beta and one is gamma. The alpha subunit is responsible for most of the biological action. It is bound with guanosine diphosphate or GDP and forms the alpha GDP unit. And the alpha subunit is also have the intrinsic enzyme called GTPase activity. The beta and gamma subunits always binds together to form beta gamma dimer, and it can also bring about the some action in the activated G proteins. Both alpha GTP unit and beta gamma dimer are united. So what happens in the membranes or in the inner surface of the cell membrane? G protein is present, and in the G protein there are three subunits. One is alpha, one is beta and one is gamma. For the biological actions, alpha is the most responsible subunit. And what is happening? Alpha subunit plus GDP makes alpha GDP subunit. And alpha subunit also have the intrinsic enzymatic activity that is known as GTPase activity. And beta gamma does what? They make the beta gamma dimer. Now, sequence of events in the formation of CAMP. First of all, hormone binds with the receptor in the cell membrane and forms the hormone receptor complex. First of all, the first point is hormone receptor complex formation. How it is formed? Hormone binds with the receptor in the cell membrane and forms the hormone receptor complex. Now, second point, it activates the G protein. It activates what? The G protein. G protein is also known as guanosine nucleotide binding proteins. And G protein releases GDP from alpha GDP unit. So G protein releases what? GDP from alpha GDP unit. Then the alpha subunit binds with a new molecule of GTP. The GTP is exchange for GTP, and this exchange triggers the dissociation of alpha GTP unit and beta gamma dimer from the receptor. Then when the alpha GTP unit and beta gamma dimer is dislocated and exchanged from the receptor, they are coming to the second messenger pathway. Now both the alpha GTP subunit and beta gamma dimer is now activate the second messenger pathway, and alpha GTP unit now activates the enzyme adenyl cyclase, which is also present in the cell membrane. Most of the adenyl cyclase protrudes into the cytoplasm of the cell and from inner surface of the cell membrane. So what it does? Alpha GTP unit now activate the enzyme known as adenyl cyclase, which is present in the cell membrane. Now the adenyl cyclase main function of this is to protrude into the cytoplasm of the cell from inner surface of the cell membrane. Now activated adenyl cyclase converts the adenosine triphosphate of the cytoplasm into CAMP or cyclic adenosine monophosphate. When the action is over, alpha subunit hydrolyzes the attached GTP to GTP by its GTPase activity, and this allows to the reunion of alpha subunit with beta gamma dimer and commencing a new cycle. So what happens when the action is over? Then alpha subunit is hydrolyzes the attached GTP to GTP by its GTPase activity, and this allows the reunion of alpha subunit with beta gamma dimer and commencing a new cycle. Now see the diagram. First of all, hormone comes, hormone receptor complex from GTP subunit is union with alpha GTP. Now from this conversion of alpha GTP and beta gamma dimer, and from this adenyl cyclase helps in this method, and ATP acts in this method, and it influences the CAMP pathway, then protein kinase A enzyme and responds to that cycle. Now go to the next part. Now we will know about the action of the CAMP. So cyclic AMP executes the actions of the hormone inside the cell by stimulating the enzymes like protein kinase A. So what the cyclic AMP does? Cyclic AMP executes the action of hormone inside the cell by stimulating the enzymes like protein kinase A. Now cyclic AMP produces the response depending upon the function and of the target cell through these enzymes. Now response produced by CAMP. So what are the responses that are produced by CAMP? Cyclic AMP produces one or more of the following responses. So number 1, contraction and relaxation of muscle fiber. Number 2, alteration in the permeability of the cell membrane. Number 3, synthesis of substances inside the cell. Number 4, secretion of or release of substances by target cell. Number 5, other physiological activities of the target cell. So what are the 5 responses? From these 5 responses, 1, 2, 3, 4, maybe something can be happened. So 1 is contraction and relaxation of muscle fiber. Number 2, alteration in the permeability of cell membrane. Number 3, synthesis of substances inside the cell. Number 4, secretion or release of substances by target cell. And number 5, other physiological activities of the target cell. Now, we have known about the main secondary messenger, that is the CAMP. Now, other secondary messengers are, in addition to CAMP, some other substances also act like second messengers for some of the hormones in target cell, like calcium ions and calmodulin. So many hormones act by increasing the calcium ion, which functions as second messenger along with another protein called calmodulin or troponin C. Now calmodulin is present in smooth muscle and troponin C is present in skeletal muscle. So calcium calmodulin complex activates various enzymes in the cell, which cause the physiological responses. Common enzymes activated by calcium modulin complex is myosin kinase in smooth muscle, and myosin kinase catalyzes the reactions resulting in muscular contraction. And in the skeletal muscle, calcium ions bind with the troponin C similar to calmodulin. Now number 2 is inositol triphosphate. Inositol triphosphate IP3 is formed from phosphatidylinositol biphosphate PIP2. So one is IP3 and one is PIP2. That is inositol triphosphate or phosphatidylinositol biphosphate PIP2 and IP3. So hormone receptor complex activates the enzyme phospholipase, which converts the PIP2 into IP3, and IP3 acts on the protein kinase C and causes the physiological responses by the release of calcium ions into the cytoplasm of the target cell. Now, diacylglycerol. Diacylglycerol or DAG is also produced from PIP2. It acts via protease kinase C. So one is calcium ions and calmodulin, one is inositol triphosphate, one is diacylglycerol. And number 4, cyclic guanosine monophosphate or CGMP functions like CAMP by acting on protein kinase A. So let's see the diagram. Hormone plus receptor makes a hormone receptor complex which will enter into the cell and cytoplasm and then nucleus. It acts upon DNA, then pre-mRNA formed. From this mRNA formed, this will come out of nucleus, and mRNA will further go to the ribosome and synthesize the protein and acts upon the response which is needed. Now the third method, that is by acting on genes. Thyroid and steroid hormones execute their function by acting on genes in the target cell. So this is the method of acting of gene. So what happens there? Hormone and receptor come, HR complex formation, HR complex acts on DNA, then pre-mRNA, then mRNA, then ribosome, then protein, then response. This is the mode of steroid hormones, and thyroid hormones are also acts in the similar way, but their receptors are present in the nucleus. So sequence of events present in the activation of genes, that is hormones enter into the interior of cells and bind with the receptor in cytoplasm or steroid hormone or thyroid hormone. So hormone enters into the interior of the cell and binds with the receptor in the cytoplasm. For steroid hormone, it is present in cytoplasm, and for thyroid, it is present in nucleus. So there is two. One is if the hormone is thyroid, then it is present on the nucleus. If the hormone is steroid, it is present in the cytoplasm. And when these are formed, they form what? Hormone receptor complex. So first of all, formation of hormone receptor complex. In case of steroid, it is formation in cytoplasm. In case of thyroid, it is processes in the nucleus. Now hormone receptor complex moves towards the DNA and binds with the DNA, and this increases the transcription of mRNA. The mRNA moves out of the nucleus and reaches the ribosomes and activates them. And when the activated ribosome produce large quantities of proteins, these proteins produces physiological responses in the target cell. So if we repeat one time the sequence of events during the activation of genes, then first of all HR complex formation, that is in case of steroid it is in cytoplasm, and in case of thyroid it is in nucleus. When the HR complex is formed, it comes towards the DNA and binds with the DNA, and when it is binds with the DNA, it will transcript mRNA, and when the mRNA is moves out of the nucleus and reaches to the ribosome and activates them, and where the activated ribosome will act upon what? Activated ribosome will produce large quantities of proteins, and when the large quantities of proteins are produced, they act upon the target cell and the physiological responses are occurring. So this is today's topic, the mechanism of hormonal action. We have discussed about all three methods in very deep, and if you have any query or any doubt, please comment down in the comment section, and do like, share and subscribe to my channel for supporting me, and thank you so much for watching.